Okay, so welcome to this next video in the playlist on experimental techniques. In this video, what we're going to talk about is the Western blot. Okay, so we're going to start off by discussing the motivation for the Western blot. What are we actually trying to do? And then we'll discuss how you would actually do it and the theory that underlies it, basically. Okay, right. So, let's start off then with the motivation for the Western blot. So basically, this is the motivation. If you have a mixture containing many different proteins, okay, so let's say we have some container here, some beaker, which contains some fluid here that contains a whole mixture of different proteins. So let's say we have protein A here, protein B here, which we'll draw as a triangle, and we'll have one more, protein C. So we won't go any further than that, but of course there could be a huge number of different proteins in this solution. Now basically, we want to be able to decide whether in this mixture of proteins, protein A is present or not. So that is basically the motivation. You are given a mixture of many different types of proteins, and you want to know there is some protein that you're interested in. Let's say it's protein A, and you want to know whether that protein A is present within your mixture. And calling it protein A, that's just meant to be arbitrary naming system, basically. It's not supposed to sort of denote in any way that it's protein kinase A. It's just some arbitrary protein. Okay, so that's the motivation. You want to be able to decide whether in a mixture of proteins, a certain protein that you're interested in is present. Now, where are you generally going to get a mixture of proteins from? Well, generally you're going to get them from cells, basically. So often the question actually becomes, you've got a cell here, okay, and you want to know whether a certain protein is present within the cell. So you might want to know whether protein A is present within the cell. So let's say here is protein A, and we want to decide whether protein A is present within the cell. Okay, right. What we're going to perform is a Western blot. This is how we're going to decide whether protein A is present within the cell or not. So, we're going to start off by discussing the lysis of the cell to get the cytoplasm out, which has all of the proteins in, and produce our protein mixture, and then we'll discuss how the Western blot proceeds from there. Okay, so basically what you will start off by doing is homogenizing your cell matter. Okay, so you'll take some tissue and you'll homogenize it up, okay, to make a homogenate. Okay, so step one is going to be to homogenize your cells. Now you will need to do this in a very careful way, because you do not want your proteins to be denatured. So homogenization is the first process, okay? So you do not want your proteins to be denatured, and the problem is that within cells you have little vesicles called lysosomes, which are full of enzymes which break down proteins, basically. So we need somehow to stop the enzymes from within the lysosomes from breaking down the proteins, because as soon as we homogenize the cell, these lysosomes are going to be homogenized up, and all the enzymes within those lysosomes are going to be released, and they could, denature our proteins, break them down. So what you do is you homogenize at a very cold temperature. Well, not at a really cold temperature, but you do it at a cold temperature, a temperature which will slow down chemical reactions, basically. It will slow down reaction kinetics, okay, and hopefully stop the enzymes from being able to degrade the proteins at any reasonable speed. In addition, you also use protease inhibitors, so you'll put protease inhibitors in with your cells so that these can inhibit the proteases that are released by the lysosomes, and hopefully stop those proteases breaking the proteins down. So, both cold temperature and protease inhibitors will hopefully join forces together to stop our protein of interest, protein A, from getting from being destroyed too much, because of course you'll have multiple protein A's. It doesn't matter if a few of them are broken up, but we don't want all of them to be broken up. So hopefully these two actions will help to prevent too much proteolysis of the protein A, so that we still have a reasonable amount of it left within the homogenate at the end. Okay, now what we're going to do then is we're going to separate out the different proteins in this mixture, because now what we've got is a mixture of loads of different proteins, and we want to separate out these different proteins basically. So the way that this is going to be done is through something known as polyacrylamide gel electrophoresis. So polyacrylamide and then gel electrophoresis. Okay, and for short, polyacrylamide gel electrophoresis is usually abbreviated to PAGE for short. So PA for polyacrylamide, and then GE for gel electrophoresis, so PAGE. Right, so basically the principle of polyacrylamide gel electrophoresis is that we're going to separate out the different proteins according to their length. So according to how many amino acids are within their polymer structure. Okay, so let me explain firstly what a polyacrylamide gel is. Okay, so polyacrylamide gels are gels which have a huge amount of polyacrylamide in. Now, what is the structure of polyacrylamide? So basically it's a polymer. So let me show you the structure of polyacrylamide. So you have a repeating unit that repeats many, many times, and the repeating unit that you have is an ethylene structure with an amide group coming off it. Okay, so here is the amide group coming off the side. So that's why it's called polyacrylamide. This is the amide here. Okay, and it's a nice primary amide. And this is going to be repeated loads and loads of times. So you'll take one of these, add it onto another one, add it onto another one, and you'll continue repeating this same subunit over again to create polyacrylamide. So, a polyacrylamide gel is a gel that has a huge amount of this polyacrylamide in. And basically, if you can imagine what will happen, these polyacrylamide molecules are huge, great polymers. So within the gel, what you'll have is these large polymers forming a meshwork like so. Now, you can imagine that if you have quite low density polyacrylamide molecules, you'll have quite big gaps between the polyacrylamide molecules. Okay, so within the meshwork, the pores as they're called, which are the gaps between the meshwork of polyacrylamide molecules, those will be quite large, whereas if you have loads of polyacrylamide molecules, a very high density of polyacrylamide molecules, then the meshwork will be far more dense, and the holes that you have between the polyacrylamide molecules will be much smaller, so the pores will be much smaller. Now, you want quite a high density of polyacrylamide because you want the pores to be quite small, because the smaller the pores are, the greater is their ability to separate the different sized proteins, basically. Okay, so let's now talk about how we're going to load the protein mixture onto our polyacrylamide gel. So let's imagine that we have our polyacrylamide gel here. So what you'll have is a rectangular plate of the stuff. Okay, so if you can imagine, I'll draw a little three-dimensional picture of this. So let me try and draw the sort of glass tray that you'll have. So here's one side of it. Okay, goes on down here, like so. And it will come up to here. So you have basically a nice little glass tray here, and you're going to put the polyacrylamide gel into this tray, and you'll have a rectangular surface of polyacrylamide gel then. So what you'll have is the gel will be in this glass tray basically. So you might take it up to this sort of a level here, so you'll have polyacrylamide gel in here, like so. Okay, right, so we're now going to load our protein sample into the polyacrylamide gel, and basically what we'll do is create a little well. So we'll basically just make an invagination of the polyacrylamide gel, cut a little bit out, this is known as a well, and we're going to put our protein mixture into here. Now before we put our protein mixture into this well, firstly we have to mix it with the loading buffer, and then we have to denature it. So firstly let's have a talk about loading buffer. So basically you're going to mix your mixture of proteins with a loading buffer. Okay, and this loading buffer will contain two important molecules. Okay, so one of the things it will contain is glycerol. Now glycerol is basically just a solvent, and it's just going to help the protein mixture to slip nicely into the well, basically. So it's just to help lubricate it in, basically, and soak into the gel. Then the other thing that you're going to contain is a tracking dye, and this is bromophenol blue generally. Okay, so you need what's known as a tracking dye within the loading buffer, and the reason for this is that what's going to happen is we're going to make the proteins move through the gel, and they will move quite slowly, but the tracking dye will move also through the gel, and it will move much faster, basically. And basically this is to make sure that we don't get the contents making it to the other side of the gel. We want to stop the process before it gets to the other side of the gel, and I'll come back to this later, because if it does get to the other side of the gel, it ruins everything. Because what can just happen is, if you leave the thing on so long that the contents of the stuff that we've put in the well can get to the other side of the gel, then basically everything can just gradually get to the other side of the well. And the point of this is to separate out the different proteins. So if you leave it on too long, what will happen is they just all end up right at the end down here, because they will all eventually make their way through the polyacrylamide gel. They'll just take different amounts of time to do that. So you need a tracking dye to tell you how far the mixture has got, basically. Okay, and the tracking dye will move faster than any of the proteins. So, if you've got the tracking dye over here, it means that all the proteins are somewhere further back. Okay, so you'll stop the process when the tracking dye gets close to the edge, so that there's no chance that the proteins will have ever got to that edge. Okay, and the tracking dye that is generally used is a tracking dye known as bromophenol blue. Okay, right, so those are the two things in the loading buffer. Now what we're going to do is denature the proteins. Okay, so first process was homogenization. The second process is mixing the protein mixture with the loading buffer. And I've skipped out a little step here. You would most likely ultracentrifuge the homogenate to remove some of the bigger molecules, so that you've just basically got proteins. So you'll spin the homogenate round and round. Some of the bigger gunk will fall to the bottom and form a pellet. So hopefully all the lipid molecules, things like that, will fall to the bottom and form a gunk. Then you can just take off the supernatant, and that will be your protein mixture. Okay, so you take your protein mixture, add loading buffer then. Okay, and now what we're going to do is denature the protein. So the next step is denaturation. Okay, so the idea is that we want to break apart the tertiary and secondary structures of the proteins. So at the moment, our proteins, are polypeptides, but they're folded into elaborate shapes, basically. And this is because of interactions of amino acids in the polypeptide with other amino acids, and you have secondary structures which are based on interactions between the core amino acid structures, so the amino groups and the carboxylic acid groups, and you also have tertiary interactions which are interactions between R groups of the amino acids. Okay, so what you will do is you'll heat the proteins up. And you do this once you've added the loading buffer, so the loading buffer gets heated up as well. Okay, and heating up destroys most of the interactions. Okay, so it almost falls apart, but it doesn't quite fall apart completely, because there is one interaction that is a tertiary interaction, i.e. between two R groups, that is too strong to be broken apart by any sort of heat that you would want to subject this to. Of course, you could heat it up further and eventually it would break, but the reality reality is that you might start breaking apart the peptide bonds there, and you don't want to do that. So you heat it up as high as you can. This breaks most of the interactions, but it doesn't break one type of tertiary interaction, and that is disulfide bonds. So bonds that form between two cysteine residues in the polypeptide. Okay, disulfide bonds. I don't want disulfides. Disulfide bonds. Okay, so to break these, you also need to add quite strong reducing agents. So you would heat it up, and you'd put some sort of reducing agent to break the disulfide bonds. Okay, and then what you would have is a protein which was just in its primary structure, so it was just a polymer of amino acids. And this is the important thing: you do not heat it high enough that you start breaking down the peptide things. You don't want the amino acids to start falling apart from one another. So it maintains its primary structure, but you destroy the secondary and tertiary structure. And now what you expose this denatured protein here, this polypeptide to, is a molecule known as sodium dodecyl sulfate. Okay, and for short, sodium dodecyl sulfate is often abbreviated to SDS. Okay, so sodium dodecyl sulfate, short hand is SDS for sodium dodecyl, and then sulfate. Right, so let me show you the structure of this molecule, because its structure is quite important for understanding what this molecule is actually going to do. Okay, so basically sodium dodecyl sulfate is a detergent. Okay, now the dodecyl here means that it's a 12 carbon molecule, so I don't want to really draw out 12 carbons. So instead what I'll do is I will use a clever trick. So I'll draw a methyl group that's right on the end, okay, and then I will draw 10 methylene groups in the middle. In fact, I might put it as 11, after all it is 11. Okay, so I'll put brackets around a single methylene group and subscript it 11. And basically what this means is you have this methyl group on the end, which I've had to draw out because a methyl group is separate from a methylene group. Some people occasionally would put methylene group times 12, and then they just have a hydrogen on the end. That's okay, but more people use this sort of a notation, where they show the methyl group separate from the methylene groups. So you have a methyl group, then you have the 11 methylene groups. That takes you up to 12 carbons. This then is the dodecyl here. Now we want dodecyl sulfate, okay? So we need to add a sulfate group onto this. So what you then have is a sulfate group. Now a sulfate group consists of a sulfur atom at the centre with 2 oxygens that are double bound to the sulfur, and then 2 oxygens that are single bound to the sulfur. Okay, now this oxygen is then bound to the carbon of the dodecyl group, and this oxygen is going to have a negative charge, so it's got a single bond to the sulfur. It's also acquired an electron from some ionic means, and that is the dodecyl sulfate. So here is the sulfate group. Now we want sodium dodecyl sulfate, so that tells us now where the ionic bond is. It's with sodium, basically. So you've got sodium and the oxygen bound ionically. So this sodium has given up one of its electrons to that oxygen of the sulfate group, and you then have them interacting electrostatically. Okay, so this is sodium dodecyl sulfate, and I should just colour in the sodium as well. There we go, sodium dodecyl sulfate. Right, okay, now basically what's going to happen is you're going to add sodium dodecyl sulfate into our solution where we've got the denatured proteins, and the purpose of the sodium dodecyl sulfate is to keep the proteins denatured. We've got them denatured now, we want them to stay denatured. Okay, so this molecule is going to bind all over our denatured polypeptide because it has polar regions and it also has hydrophobic regions, so it can interact with both hydrophobic portions of the polypeptide and also polar portions of the polypeptide. Now, the sodium goes off, okay, so as soon as you put sodium dodecyl sulfate in solution, it separates into the dodecyl sulfate anion and the sodium cation, so they no longer remain bound together, and it's the dodecyl sulfate anion that ends up all over this protein. So what you end up with is, I'll just show them as circles, you end up with sodium dodecyl sulfate molecules all over the polypeptide, and these are going to maintain the polypeptide in its denatured state even after you remove the high temperature. Okay, so here we go. So there we go, there now we have our polypeptide covered in sodium dodecyl sulfate molecules, and sodium dodecyl sulfate does more than just keep it in the denatured state. It is actually going to perform a really important function, because remember, this is the dodecyl sulfate anion that is coating the polypeptide here. And the dodecyl sulfate anion is called an anion because it has a negative charge. So actually you have covered your polypeptide in negatively charged molecules. So this polypeptide is going to become negatively charged, basically. Okay, and that's going to be important. That's how we're going to get these proteins to move through the polyacrylamide gel.